My name is Robert Lawless. I'm a student at Mount St. Mary College in Newburgh, New York, and I will be discussing the article and study on effects of fragmentation on plant adaptation to urban environments. This was conducted by Jonathan Dubois and Pierre-Olivier in various locations in France. Some background: urban ecosystems are heavily altered by human existence. This affects the plants, animals, and other living organisms. Many habitats are also fragmented, which subjects these areas to higher temperatures, but not naturally. Fragmentation is often defined as a decrease in some or all types of natural habitats in a landscape, and the dividing of the landscape into smaller and more isolated pieces. As the fragmentation process develops, the ecological effects will change. Fragmentation can be caused by natural processes such as fires, floods, and volcanic activity, but is more commonly caused by human impacts. It often starts with what is seen as small and harmless impacts, but as human activity increases, the influence of fragmentation becomes greater. So, the study. Crepus sancta, known as Astercae, is an annual Mediterranean wasteland weed which occupies fragmented urban areas as well as unfragmented urban areas in southern France. The right shows a picture of the Crepus sancta. Most people know this weed as a dandelion, as you can see by looking at the picture. This specific variation of Astercae grows in France, but we do see a different kind of variation of this weed in the United States. And during this study, they tested for shifts in dispersal, reproductive traits, and size across a rural-urban gradient to learn whether and how selection may be driving changes in life history in urban and fragmented habitats. This is a map of where the many forms of this weed grows. As you can see, it's very common, and I definitely see it in many areas that I've been to. So some effects of fragmentation. Fragmentation affects both demographic and genetic processes. Fragmentation reduces the size and increases the isolation of plant populations and pollinators, which may see a decline in seed setting. Fragmentation also affects selection on dispersal, for example, reducing populations of dispersers or limiting access to suitable habitats. Dispersal in this case is referred to as the dispersal cost, which can be classified into energy, time, risk, and opportunity cost. Dispersal is often referred to as an event or a single stage, or any stage actually. The dispersers in this study would be the fragmentation, which would be the event, and the effects of this on the habitats. So some expectations to the study of fragmentation, which was observed. Increases in the frequency or intensity of disturbance to decrease population density and competitive interactions. Correlations between the phenotypes and environmental conditions would be a clue of adaptation. Although one may expect urban habitats to favor evolutionary shifts in plant species, it may be difficult to identify which environmental factors are acting as the primary effect. So this slide is basically about the areas and growth of this species and weed. Contrary to abandoned fields, plowing in vineyards prevents the development of older successional stage. Consequently, this means the density of C. sancta varies from 50 individuals per square meter in cultivated vineyards to only 5 to 10 individuals per square meter in old fields. Urban fragmented populations exhibit similarities, with abandoned fields counting only 5 individuals per square meter. The low density of C. sancta and the presence of more competitive species in both old fields and patches in an urban fragmented environment can be interpreted as an older successional stage compared with the plowed cultivated vineyards. Patches, extinction, and low interpatch connectivity in urban fragmented populations may also lead to reduced local density. Urban environments also support a few less fragmented habitats within the city, which allows us to disentangle the effect of fragmentation from the effect of other factors associated with urbanization. However, highly fragmented populations cannot be found in rural environments. So the study. Crepus sancta is a common ruderal winter annual in southern France. It is partially self-incompatible, heterocarpic, and flowers in early March. This species pops up in fragmented patches around trees. It also comes about in parking lots, and which is very different from the rural vineyards and farms that we were discussing earlier. These weeds also spring up around the concrete and asphalt patches and in grassy areas, like we see in our region. This also happens in the European regions. The tests will include different populations of the species and will be grown in a garden under certain circumstances. Now I'm going to discuss the garden experiment that was conducted. So they collected several ripened inflorescence and maternal leaf tissue from 30 to 40 plants in each of 6 populations. Of the 6 populations, 2 were rural and 4 were urban, and they represent the rural and urban populations with letters, which is basically where they got them from. They first selected on the basis of studies to maximize the range of habitat fragmentation, and 2, to discriminate the effects of urbanization from fragmentation on selection for life history traits. So furthermore, on the experiment, the 2 rural populations occurred within large, unfragmented habitats from Montpellier. CL occurred in a vineyard with 50 people per square meter. PSL occurred in an abandoned vineyard of about 5 years with a population of 10 people per square meter. The urban populations, 2 of the 4 were unfragmented from a dense lawn and vineyard in the city. The other 2 urban populations were highly fragmented with 5 to 10 people per square mile. So during the experiment, this is what they were looking at and how they took the measurements and the data. So they took photographs and measured the spore shape and the plant and leaf size. They recorded the date of the first capitulum emergence and its diameter and length of its first flowering stalk, which is basically like when the yellow flower came out and it sprouted out. And they also facilitated pollinations from hives in the greenhouses to, make it a more natural environment, which would occur if they were in the fields or in the vineyards or wherever they were. And during the experiment, they also used markers to access levels of genetic diversity within each population between fragmented and unfragmented, and they extracted the DNA using leaf tissue. So some results and conclusions to the experiment was that the mean number of alleles per population and per locus varied from 5.5 to 17, providing good statistical power for detecting genetic structure. Most populations showed a small deficiency of heterozygotes, revealing inbreeding, and the PCA sample revealed that two independent components captured most of the total phenotypic variability, accounting for 71.7% of the total phenotypic variability. And here is a good visual of the correlation between the measured traits and this shows the different samples that they took and how large the flowers got between the unfragmented and the fragment versus the rural and all that, and then they have it all labeled with the lines. And in this figure here, this shows the mean and standard error for the ratio of non-dispersing seeds to the total seeds, and the different samples are in different color lines here as well. So some discussions which I felt was important, which was written by the authors. The study examined patterns of differentiation and putative adaptation to urban environments among three groups of plant life history traits, one being the reproductive traits, two being the size traits, and three being the dispersal traits. We further sought to distinguish urban effects from the effects of the habitat fragmentation in urban areas, while size traits variations is not linked to the degree of habitat fragmentation. We found that habitat fragmentation accounts for differentiation of dispersal traits for these populations. To a lesser extent, reproductive traits exhibit a smaller pattern, although one population did not conform to this scheme. By contrast, urbanization showed no effect on phenotypic trait variations. And then here's the works cited.